Alopecia areata is an autoimmune form of hair loss where T cells attack the growing hair follicle. It is not a life-threatening form of autoimmunity. However, it is a life-altering form of autoimmune disease. It affects patients in a very profound and very personal way to undergo hair loss. I could say that with authority because I'm one of them. So the reason my lab studies this is because I actually have alopecia areata, and that's what really sparked us to do this type of work. Over 6 million people in the U.S. also suffer from various forms of alopecia areata, so this is a tremendously large and motivated population of patients who have no other treatments available.
Treatment for this disease in the past has been hit and miss with steroids and with varying degrees of success. We've been excited to see data both in mice and now in people to show that we can reverse this disease with small molecules that are aimed at reversing the inflammatory condition in humans.
The trial for ruxolitinib in the treatment of alopecia areata got started based on the genetic and immunologic discoveries made by Drs. Cristiano and Kleins. In investigating the genetics of alopecia areata, they were able to then subsequently pinpoint several immune pathways that appear to be important in alopecia areata. The next step was identifying available drugs that actually inhibit these pathways, and this is the basis upon which ruxolitinib was selected. Once the drug was selected, we then went about designing a trial to preliminarily test its effect on alopecia areata.
We now have the ability to identify potential targets for treatment, and using what some people call a mechanism-driven approach, that is to look for pathways that are altered in human disease, and then to see if those alterations lend themselves to some type of manipulation, either pharmacologically or genetically or both. And I think that's what we have in the case of this study, which is an autoimmune disease that has eluded for decades any basic understanding of the development and the causation of the disease.
We went on to show which immune pathways might be important in the cell death of the hair follicle, and then using those insights, we blocked those pathways in the mouse models, showed they were efficacious, and then went on to try medically relevant antibodies and small molecules to see whether when we block these pathways we could reverse alopecia areata. And we showed that it was true in mouse models, and then went on to go to a small series of patients and show that the therapies could also be effective in people.
We are very committed to making sure that that patients with alopecia areata have access to this class of drugs. Hopefully in a way someday that's not an off-label indication, meaning we'd like to pursue getting a label, getting an indication for a JAK inhibitor in alopecia areata. We're deeply committed to doing that. There are so many other diseases, if you will, ahead of us, other diseases that might be considered much more life-threatening or life-altering, that alopecia can sometimes get pushed to the back burner or to the bottom of the list. But this has been the major focus of our entire group, and we would like very much to make sure that this ball gets across the finish line, whatever that takes. And hopefully we'll be joining with some pharmaceutical partners to make sure that alopecia stays top of mind and that someday this is an approved indication for JAK inhibitor.